Our lecture today will be about body energetics, metabolic rate, and regulation of food intake. So we have seen whatever we are eating finally is digested by the activity of digestive enzymes. Then we have absorption, and we have foodstuff are reaching our metabolic pool. And we are using this foodstuff from our metabolic pool as a fuel for our body energetics. So we have cells which are using this foodstuff to generate macroenergetic molecules like ATP, and then we have in our body a lot of works that are taking place. They are consuming this ATP, and we are using this ATP to perform body works. In our body, we have plenty of works like chemical works, which are consuming macroenergetic molecules for building of cellular components, secretion, synthesis of enzymes, and so on. Also, we have plenty of mechanical works. We have muscle contractions, heart pumping, intestine is moving, and so on. Also, we have plenty of electrical works. We have generation of action potentials, all excitable cells. We have nerve conduction. We are maintaining resting potentials. All these activities are maintained by the activity of pumps, which are regulating the amount of sodium and potassium across the cellular membranes. We have, as you know, keeping low concentration of sodium inside. We are keeping also low concentration of potassium outside cells or in body fluids, which is important for the electrical work that is performed in excitable cells. As we see from this slide, we have a lot of metabolic activities that are taking place in our body, and these metabolic activities to generate macroenergetic molecules which are useful for our body work. The question is, can we measure all these metabolic activities that are taking place in our body? As metabolic activities, as you see, we are using foodstuff finally to generate macroenergetic molecules, but we have end products by the metabolic activities that we are having. We have final burning of this foodstuff results in generation of water and CO2. Also, NH3 can be generated by the catabolism of amino acids. As we see, foodstuff from fat, carbohydrates, and proteins are undergoing degradation by our metabolic system, and by this process of degradation, we are generating macroenergetic molecules. The bulk of the generation of macroenergetic molecules is taking place at mitochondria. At that organelle, we have electron transport system, we have oxidative phosphorylation process, which are finally releasing bulk of macroenergetic molecules, and as end products by this final burning, we are getting also generation of CO2 and water. So we have in our body a system for processing foodstuff to get macroenergetic molecules. We have an anaerobic system and we have aerobic system for generation of macroenergetic molecules. By an aerobic system, we are generating intermediate molecules and we are not getting the final degradation of these foodstuff to their end products. But by this system, we are generating small amounts of macroenergetic molecules. Then we have the mitochondria, the site where we have the aerobic system for generation of macroenergetic molecules. At that level, we have the bulk for the generation of macroenergetic molecules. As you know, at that level, we have oxidative phosphorylation enzymes, we have the oxygen or the electron transport system, and that system is consuming oxygen molecules for further burning of the intermediate products, which is a chemical burning, and we are getting finally generation of more amounts of macroenergetic molecules. In addition to that, we are also generating carbon dioxide and water as end products. So as you see, we have oxidative phosphorylation system at the level of mitochondria, where we are consuming oxygen and generating carbon dioxide as end product. Now we have a term which is related to that metabolic activity, which is called respiratory quotient, which refers to the volume of carbon dioxide produced per unit time to the volume of oxygen consumed per unit time. By calculating respiratory quotient, we can have an estimate about the main fuel of foodstuff for our body energy. As example, for final degradation of one glucose molecule, we are consuming 6 oxygen molecules, and by final degradation, we are generating 6 carbon dioxide molecules. As a result, if we are calculating the respiratory quotient for glucose will be around 1. If we have fat as fuel for our body energy, respiratory quotient will be around 0.7. If we have proteins or mixed food as fuel for our body energy, respiratory quotient will be around 0.82. Returning back to our question, if we can estimate the metabolic activities that are taking place in our body. As an answer, we can measure the metabolic rate, which refers to metabolic activities that are taking place in our body per unit time. During metabolic activities, we are generating macroenergetic molecules. And by using these macroenergetic molecules in our body works, we are finally generating that energy into heat. As you know, energy cannot be destroyed nor created, but can be transformed from one form to another. So, by final conversion of the energy which have been used for our body work into heat, and by measuring the amount of heat irradiated from the body, we can have an estimate about the metabolic activities that have taken place in that body. By measuring the amount of heat produced by a body per time unit, we are measuring what we are calling the metabolic rate. So as units for measurements of metabolic rate, we have it in the form of calories per hour. Pay attention to calories with the capital C, which refers to kilocalories. How we are measuring the metabolic rate? We have direct methods for measurement of metabolic rate, and we have indirect methods for measurement of metabolic rate. For measuring the metabolic rate by direct method, we are placing the person in an insulated room. That room is equipped with a water flow. Now, if you know the temperature of the water which is flowing inside that insulated chamber and know the temperature of water at the outflow, and you know the flow rate of water, you can calculate the amount of heat that has been accepted by water flow through that installation system, which is exactly equal to the amount of heat that has been irradiated from the body. The second method for measuring the metabolic rate is by indirect method. This method is based on the amount of oxygen consumed per unit time. By measuring the rate of oxygen consumption per unit time, we can proceed with the calculation of metabolic rate of that body. For each liter of oxygen consumption, the body is irradiating around 4.8 calories. This is called energy equivalent of oxygen. As an example of calculation, if we have 1 liter of pure oxygen consumption per 5 minutes, that means we are having 12 liters of pure oxygen consumption per hour, and the metabolic rate is a result of multiplication of 12 liters by 4.8, which is the energy equivalent of oxygen, which will be equal to 57.6 calories per hour. Measurement of oxygen consumption per time unit can be by closed methods and open methods. Closed method can be done in the lab with the help of a device which is called Spirometer. That device is usually used for measuring respiratory volumes. This device is composed of an inverted drum over water bath. Inside that inverted drum we are placing air. If we had placed, for example, 10 liters of atmospheric air and adding 2 more liters of pure oxygen, now we have 12 liters inside the device. Of these 12 liters, we have 4 liters of pure oxygen. And we have created a closed system with a line. In that way, this person is inspiring air from inside the spirometer, while in the expired air is going back to the spirometer, but in the way of airflow out, we have another device by which we can get adsorption of the CO2. We are placing usually some chemical substances like soda lime to absorb the CO2 which is expired from that person, and we have the returning back only the air without the CO2. Now, with time, to say after 10 minutes you are not finding 12 liters in that device. So with time, the amount of air in that device, to say after 10 minutes, was 10 liters, which means there was a consumption of 2 liters of oxygen by that person. Then, by knowing the rate of oxygen consumption per 10 minutes as 2 liters, we can proceed with the calculation of metabolic rate for that body. The second method for measuring oxygen consumption is called open method. In that method, the person is inspiring atmospheric air. You know, the oxygen concentration in atmospheric air is around 20%, while the expired air is collected into a bag. Once you have, to say, collected 100 liters of expired air in that bag, you are assaying the concentration of oxygen in the expired air. You are not finding the same concentration of oxygen as inspired air. So to say, after 15 minutes, you have collected the 100 liters of expired air, and in that expired air, you have found that the concentration of oxygen is 17%. So we have a difference of 3%. By that difference, we can estimate the amount of oxygen consumed. Now you know that in 15 minutes, that person has consumed 3 liters of pure oxygen. Then you can proceed with the calculation of the metabolic rate for that person. To standardize our calculation, we are relating the metabolic rate to the surface area of the body. As an example, by our calculation, we have the metabolic rate is 57.6 calories per hour. This person, to say, is having a surface area of 1.6 meters square. So, by calculation and relating it to 1 meter square surface area, we have divided the 57.6 over 1.6 meter squares, and we are getting finally the metabolic rate of that body is 36 calories per hour per meter square, which can be written also as calories dot hour to the minus 1 dot meter to the minus 2. Also, for more standardization of our testing, we can measure the metabolic rate under basal conditions. By measurement of metabolic rate under basal conditions, we are now talking about measurement of basal metabolic rate of that body. These are the basal conditions under which we are measuring to get basal metabolic rate. No eating food for at least 12 hours before measurement. If somebody has eaten, for example, there is an increase in the gastrointestinal activity, and this may affect the oxygen consumption by that body, and as a result affecting our measurement. Secondly, measurement after a night of restful sleep. During measurement, this person must be awake, not sleeping. Third, no exercise in the hour prior to the test. If somebody had some exercise before measurement within 1 hour, there is a consumption of oxygen stores from the body. Then the person is needing at least 1 hour to replenish the oxygen consumed from stores. This will affect our measurements also. Elimination of all factors that may cause excitement. During measurement, that person must be calm, not excited, not stressed, not thinking about any problem that may increase metabolic activities in that body. Last, measurement at comfortable temperature. If we are measuring at lower temperature, for example, the metabolic rate in that person is higher. To regulate body temperature according to the ambient temperature, this will affect also our measurement. We have a lot of factors that can affect the metabolic rate of the body. Don't forget, here we are talking about the metabolic rate and not the basal metabolic rate. With exercise, there is an increase in metabolic rate. According also to daily activity, the same person can have lower or higher metabolic rate. You are staying on a chair, for example, doing nothing, you are having lower metabolic rate than a person who is doing heavy work. With age, there is a decrease in metabolic rate. Older persons are having lower metabolic rate than kids. Don't forget, we are relating the metabolic rate to meter square surface area of that body. With sleep, the metabolic rate is lower than the metabolic rate of our awake person. Also, according to climate, we can have differences in metabolic rate. Persons who are living in cold climate, they are usually having higher metabolic rate than persons who are living in hot climate. With fever, there is an increase in the metabolic rate. There is a huge amount of heat radiated from that body, also higher oxygen consumption for their metabolic activities, so the metabolic rate in fever condition is higher than in the healthy person. With malnutrition, there is a decrease in the metabolic rate. Actually, the body is trying to conserve the use of food stuff for their body energetics, so the metabolic activities are reduced. We have also what we are calling specific dynamic action. What we mean by that, it has been observed that persons who are placed on protein diet, they are having higher metabolic rate than persons who are on carbohydrate or on fat diets. So this has been explained as we have a specific action of some amino acids to increase the metabolic activities in that body, and they have called these specific actions as specific dynamic action of some amino acids. Also, the hormonal level in the body can affect the metabolic rate. Now, when you are taking the endocrine system, you will see that we have what we are calling metabolic hormones. For example, an increase in thyroid hormones can increase the metabolic rate. Also, sex hormones can increase the metabolic rate. But the increase by male sex hormones is higher than the increase by female sex hormones. Also, the level of growth hormones can increase, higher level of growth hormones can increase the metabolic rate. Also, the metabolic rate can be affected by the sympathetic tone in the body. During stress, for example, or fight or flight reaction, there is an increase in metabolic activity, and accordingly there is an increase in metabolic rate. The last subject we are talking about is regulation of food intake. Every day we are ingesting certain amount of food. That food is having energy value. As you know, the energy value of 1 gram of carbohydrate is around 4 calories, and for fat is around 9 calories, and for protein is around 4 calories. Also, every day we are consuming macroenergetic molecules to cover our daily activities, and we are generating specific amount of heat, which is our metabolic rate for that day. So this is called as energy expenditure. Usually the energy value of eaten food is exactly equal to the energy expenditure for that day. So a healthy person, all the time he is in what we are calling neutral balance. If the energy value of eating food is more than the energy expenditure, we are talking about a person in a positive balance. If the energy value of eating food is less than the energy expenditure for that day, we are talking about a person in a negative balance. Usually we are in a neutral balance. Now, the question is how your body knows how much food to eat with energetic value that are covering exactly what is needed for energy expenditure for that day. In hypothalamus, there are feeding centers, which are represented by lateral nuclei. By stimulation of feeding centers, there is an increase in feeding behavior. Also, there are satiety centers, represented by ventromedial nuclei. By stimulation of satiety centers, there is a decrease in feeding behavior. In addition to these neural structures, there are also other structures which are involved, like amygdala. It was found that the destruction of amygdala results in what we are calling psychic blindness for the choice of food. These persons are eating anything they are seeing. In addition to amygdala, also prefrontal cortex is also involved. The prefrontal cortex is mainly involved in stimulation of appetite for certain types of food. So as we see that the main centers which are involved in the regulation of the amount of food, they are feeding centers and satiety centers. We have a lot of signals that are coming from the body to change the activity of these centers. By changing the activity of these centers, we are feeling either hungry or we have the feeling of satiety. So as we see, we have a lot of neural signals which can change the activity of feeding or satiety centers. These signals are coming from the body through the vagus nerve. Also, we have a lot of hormones which are involved in changing the activity of feeding and satiety centers, like leptin hormone, insulin, ghrelin, polypeptide, YY. As you remember, we have talked about group of hormones released along the gastrointestinal tract by endocrine system there, and for sure some of these hormones are involved in the regulation of food intake. So as we see, we have also a lot of hormones, glucose level, for example, signals from the gastrointestinal tract, signals from the adipose tissue that we are having, also from the adrenal glands. All of them, they can change the activity of these centers. To understand this complex process of regulation, we can divide the regulatory mechanisms that we are having for the activity of these centers as short-term regulatory mechanisms. Also, we have long-term regulatory mechanisms which are involved in changing the activity of hypothalamic centers. Long-term regulations, we have many theories which are explaining the process of regulation of the activity of these centers. We have glucostatic theory, which is stating that the level of glucose in body fluids is a stimulus to eat or not to eat. That theory in this way is not right, actually. As you know, diabetic patients are having plenty of glucose in their body fluids, but also they are having the feeding is stimulated. So they are generating what we are calling polyphagia or hyperphagia. They want to eat more. That theory has been changed to the availability of glucose for cellular energy is the stimulus to eat or not to eat. In that way, that theory has been corrected actually. In this case, in diabetic patients, they have plenty of glucose in their body fluids, but that glucose is not available for cellular energy. Lipostatic theory is talking about fat deposits. The increase in fat deposits in our body is the stimulus for these centers to reduce feeding behaviors. Increased fat content in adipocytes results in increasing expression of leptin hormone. That leptin hormone is acting over hypothalamic centers to reduce feeding behaviors. The leptin hormone is produced by a gene which is called OB gene, which refers to obesity gene. Any defect in the OB gene may result in a non-functional leptin which is not having optimal activity over the feeding centers and is not having effect over the feeding behaviors. Other theories which are involved in explaining the regulation of food intake, we have aminostatic theory, which is talking about the level of amino acids in our body fluids, the stimulus to eat or not to eat. Also, we have the activity of thermoregulatory centers. We have a strong relation between the activity of these centers and feeding centers. You are thinking well in winter, you are eating more than in summer. The activity of thermoregulatory centers in winter is higher than in summer. In addition, we have also psychosocial factors could be involved. Some people are accustomed to eat 3 meals a day. So once the time of the second meal or the third meal, they are feeling hungry. Other people, they are accustomed to eat only 1 meal a day. If they are missing the time of the second or the third meal, they don't care about the feeding at that time. In addition to long-term regulatory mechanisms, we have also short-term regulatory mechanisms which are involved in controlling feeding behavior. First, we have gastrointestinal filling. Every time you have filled your gastrointestinal system with food, you have a lot of signals, neural signals and hormonal signals that are acting over these centers to reduce feeding behavior. In addition, we have hormones also. I think you know we have a lot of hormones released along the gastrointestinal cells by endocrine cells of the gastrointestinal tract. For sure, some of these hormones are acting over these centers and changing their activity. We have hormones like insulin, for example, cholecystokinin. They can reduce feeding behavior. In addition, we have also another theory which is talking about having receptors at the upper part of the mouth and at the fronts which are metering the amount of food that we are eating, and they are sending signals toward the hypothalamic centers to reduce feeding behaviors. Obesity is considered as an increase in the body weight by more than 10% of the ideal body weight. But that increase in the body weight is by deposition of more fat in their body. Athletic persons, for example, they have an increase in the body weight. But the increase in their body weight is by increasing muscle mass. We are not considering them as obese persons. Now, what we mean by ideal body weight, it is related to the height and sex, but generally we can calculate or estimate an ideal body weight for a person according to his height. If a person is having 170 centimeter height, for example, his ideal body weight is around 70 kilograms. Many factors could be involved in the pathogenesis of obesity. I think you know now we have hypothalamic centers and their activities depend on the stimuli that are coming from the body. In some people there is a need to stronger stimuli to inhibit feeding behavior. Some others they are needing weaker stimuli, and by these weaker stimuli they are getting inhibition of feeding behavior. So we are not having the same level of activity for the whole population. Some persons they are needing stronger stimuli to inhibit the feeding behavior. So these persons are subject to develop obesity. In addition, we have genetic factors could be involved. You know that some obesities are running in families. If the father is obese or the mother is obese, most likely to have their kids also obese. One thing that we have seen is the OB gene, which is the gene which is producing leptin. If there is a defect in the OB gene, these persons are expressing a non-functional or a defective leptin which cannot act well over the hypothalamic centers, and they are subject to have increased feeding behaviors and more deposition of fat in their body. Psychosocial factors also are cause of obesity. You have friends, and once you are visiting a friend, he will serve you with a lot of food, beverages. What happens to people who are all the time visiting and visiting? They are consuming more than what is necessary for their body energetics, and they are depositing more fat in their body, and they are subject to develop obesity. Also, childhood overnutrition could be a cause for the generation of obesity. That overnutrition during the childhood period may result in increasing the number of fat cells in their body. Now, once they are getting bigger, they have to fill these fat cells to start releasing good amount of leptin hormone to reduce feeding behavior. So, that overnutrition during the childhood period could be also a cause in the pathogenesis of obesity. Other causes for generation of obesity, disorders of the endocrine system. As an example, in hypothyroidism, there is a decrease in the metabolic rate of the body. If these persons are consuming the same amount of food as a healthy person, they are subject to deposit more fat in their body and to develop obesity. Also, by the lack of exercise in a healthy person, there is a decrease in the metabolic rate of that body. If these persons are also consuming normal or same amounts of food, they are also subject to develop obesity. In addition, is having a body weight less than the ideal body weight by more than 10%. These persons are in a negative balance. So the energy value of eating food is less than what is necessary for energy expenditure that are covering their daily activity. There are many causes for inanition. Some of them they are related to the activity of the hypothalamic centers, but also we can have psychogenic causes. Persons who are depressed, for example, they are subject to develop what we are calling anorexia nervosa. They are eating less than what is necessary for their body energetics, and they are becoming in a negative balance. As hypothalamic abnormalities, as you remember, we said we have a specific level for the activity of hypothalamic centers to regulate the food intake. So some people, we have there is a need to weak stimuli to inhibit feeding behavior. So these persons are subject to eat less than what is necessary for their body energetics and to develop inanition. The last subject we are talking about is starvation, which refers to have no food available for body energetics. So, in that condition, there is a depletion of stores from the body. The first stores which are depleted are carbohydrate stores. Within 1-2 days, there is consumption of all carbohydrate stores from the body for body energetics. Then, there is a decrease in fat and protein stores. There are consumption of these stores which take place for about 1 week. Then we are continuing with consumption of fat stores and trying to conserve protein stores. As you know, most of these proteins are functional proteins, and the body is trying to conserve the use of proteins for body energetics. So they are consuming more fat for body energetics. This happens and taking place for about 6 weeks. After 6 weeks, there is high depletion of fat stored from the body. So the body now is returning to protein stores to consume these stores for body energetics. So there is now an increase in the depletion of the protein stores. If you are viewing the protein stores, the depletion of these stores is taking place in 3 phases. The first week, there is high depletion, then followed by lower depletion of protein stores. But after week 6, there is also, when the body is returning back to protein to use them as source of energy, there is an increase in the depletion.
Try to relate the period at which there is high consumption of fat for the body energetics with respiratory quotient. As you see, the week 2, 3, 4, there is high consumption of fat, and the respiratory quotient in these persons actually will be closer to the respiratory quotient when we have fat as a source of energy, which is around 0.7.
Thank you and good luck.